Howdy, my name is Stephanie Aranda and I'll be presenting the Acute Gastrointestinal Disease by Noroviruses. This subject can be found in Chapter 12, Family Calciviridae, in Susan Payne's Viruses from Understanding to Investigation book. Introduction. The Centers for Disease Control and Prevention, known as the CDC, has established that their estimates show that half of all food-related illnesses outbreaks are caused by noroviruses. These outbreaks often occur mostly when people gather or when food is served. The family of the virus particle that causes this acute gastrointestinal disease is the family Calciviridae, with the classification of a genus norovirus. We'll be going over the basic information of the norovirus that causes acute gastrointestinal disease. One of the first things to know is that those who have to be cautious of this disease are mostly children and the elderly, as it can be serious for them. Norovirus infections are not a surprise in a hospital or other healthcare facilities for treatment, as this virus is very contagious and is hard to eliminate when unnoticed. Many people, especially the uneducated, often think that any disinfectant can kill all viruses and bacteria within the given spot that is treated. The CDC recommends that for proper sanitation of surfaces and utensils is to utilize a chlorine-based product or sanitizer approved by the EPA for use against the norovirus. This figure displays, as of June 22, 2021, the EPA's registered antimicrobial products effective against the norovirus. This document would be most helpful for those susceptible to the severe side effects of the norovirus. We will be discussing the viral genome of the norovirus along with its qualities of the particle. VP1 is a complex structural protein of the capsid that it also appears to play a fundamental role in its interaction with the human norovirus particle with the whole cell, and is essential for classification of these viruses. The VP2 is a minor structural protein that is not studied often. Studies have reported that this is not essential for the formation of the VLPs. The VLPs are often formed by interactions between capsid proteins to form the final structure. The VPG is a viral protein genome linked, which is a non-structural protein that is connected with a positive sense single-stranded RNA, which is the ssRNA+. The poly-A tail, which is the polyadenine tail, is located at the 3' ends of the positive sense RNA of the genome. We will be discussing the viral and genome replication cycles of the norovirus. The norovirus attaches the cell using its surface proteins to allow penetration for the genome release in the cytoplasm. The replication cycle takes place in the cytoplasm and is considered cytoplasmic, allowing the positive strand RNA virus to translate utilizing the non-structural proteins that include the protease and the RDRP enzyme. The VPG protein is then utilized for supporting the translation roles and initiating factors. With the icosahedral symmetry of T equaling 3, the capsid assembly requires 290 copies of VP1 and a few of the VP2. Thus, the last step is the infected cells undergo lysis to release the viral particles. Okay, for transmission, noroviruses are resistant to common disinfectants and have a good chance of survival and transmission via indirect contact through fomites. Fomites are objects that contain viruses on surfaces, as some have the stability to stay alive. Fomites may be in contact with food that you eat or with the hands that get in contact with your face. As you can see here, the green highlighted people or sections of the body are showing how the norovirus could be transmitted. The pathogenesis of the norovirus is affected by many factors when crossing the barrier. Factors that could include the presence of the bacteria, which is A, the absence of bacteria and altered innate immune responses, which is shown as in B, the binding of the enteric bacteria of B cells, which is on C, the infection of the macrophages to utilize in penetration, which is seen on D, and lastly, on E, the infecting the cell directly to trigger expression to allow the spread of infection. For disease manifestation, the many food-related illness outbreaks are recorded and collected by the CDC and are due to the noroviruses being highly contagious and stable on contaminated surfaces to allow indirect infection through the result of ingesting contaminated food or water. This often results in physical symptoms of nausea, diarrhea, and stomach pain. For diagnosis, diagnosis of the norovirus is mostly based on symptoms presented that are often seen as the stomach flu. The norovirus is associated with short-term diarrhea and vomiting if the infected person is immunocompromised or has any illnesses of concern, then the doctor may recommend a stool test. The stool test is done through a rapid immunochromatographic assay to detect the presence of the norovirus antigen in the human fecal sample. These are the resources I have utilized in preparing this presentation. I have used Susan Payne's book, Chapter 12, Family Calciviridae, as my most used resource when collecting information. And this concludes this presentation of the norovirus causing the acute gastrointestinal disease. Thank you for viewing this presentation. Have a great day.